Good morning, and thank you, Emmett and OIS, for inviting us to speak today about AsclepiX Therapeutics. As we know, anti-VEGF monotherapies have really hit a ceiling for efficacy, and they have a relatively short duration of action. AXT107 may be a best-in-class agent because it inhibits VEGF, it activates Tie2 as a monotherapy that may be dosed once per year. This may dramatically reduce the treatment burden for patients. AXT107 has demonstrated superiority and longer durability to Eylea and preclinical animal models, and has demonstrated excellent safety and tolerability in toxicology models for 15 months to date and in 2 species. AXT107 has a very unique mechanism of action. It strongly inhibits VEGF, and it activates Tie2 as a monotherapy, one molecule that affects the 2 proven pathways of disease. And it does it by binding to the integrin co-receptors. So alpha V beta 3, it binds to that with the VEGFR2 receptor. It inhibits VEGF-A and VEGF-C, and it completely stops vascular leakage and neovascularization. On the other side, it binds to alpha 5 beta 1, the co-receptor for Tie2, and there it uses the ligand Ang1 and Ang2 to superphosphorylate Tie2 to also stop vascular leakage. And let's take a look at the preclinical data. Here you can see that AXT107 has demonstrated superior efficacy to Eylea with longer duration of action in this rabbit VEGF-induced vascular edema model. Both drugs are dosed on day 1, but you can see that they both reduce statistically significantly leakage on day 30, and that's after 10 micrograms of VEGF are injected at day 23. By day 60, with another injection of 10 micrograms of VEGF, AXT107 is still reducing that leakage where Eylea is inactive. AXT107 also inhibits VEGF-induced retinal detachment in this TED-opsin genetic mouse model. In this model, you can see that with 30 times the level of VEGF, the Lucentis and Avastin work, but there are a lot of total retinal detachments. Eylea works much better in this model, but there's still a total retinal detachment and a partial detachment. But AXT107 had no retinal detachments and only one partial detachment that actually resolved. AXT107 also causes a regression of the neovasculature. In this laser-induced treatment model for wet AMD, the animals are given a laser, divided into 2 groups, and you can see that in the placebo group, that continues to grow as it does in disease. But when AXT107 is injected, it actually results in regression of the neovasculature. It may have the potential to turn back the clock on this very difficult to treat disease. AXT107 also inhibits Ang2-induced vascular leakage in vivo. This is a genetic model where Ang2 overexpression is found in the mouse. As you can see, normal vessels where no Ang2 is elevated are very tight and not leaking. But with high Ang2, there is a lot of leakage on the right panel, and this is true as in disease. But when AXT107 is injected with high Ang2, that leakage is stopped. And this is due to its excellent work in the pathway of phosphorylating Tie2. Now, we said that it may be dosed once per year, so AXT107 forms a clear gel upon injection in the eye. It stays right where it's injected, below the visual axis. It does not block the optic nerve, doesn't break apart, and this is in 15 months of animal data. So it provides maximal efficacy, very good properties that allow strong efficacy and safety with potentially dosing once per year. We've demonstrated that AXT107 100 microgram and 500 microgram is efficacious for 13 months to date with a single injection. And here you can see in this rabbit VEGF-induced vascular edema model, the dose, one single dose of AXT107 is dosed to the animal on day 1, and day 23 of each month It's injected with 10 micrograms of VEGF to induce leakage. And you can see that AXT107 as a monotherapy overcomes that leakage very, very strongly and potently for 13 months to date. Here's our clinical timeline and our milestones. Currently, we are completing our IND-enabling tox program, plan to submit our IND in mid-2020 and start our phase 1/2 clinical trial for DME in next year, followed by wet AMD. We have a very strong team of investors and have just recently added to our board of directors Stephen Altschuler, MD, the founder and chairman of Spark Therapeutics. We're very excited to bring this product to help patients with retinal diseases where we strongly inhibit VEGF, we activate Tie2 with potentially 1 dose per year to preserve vision with patients who have retinal disorders. Thank you so much.